How does H. pylori cause stomach cancer? Helicobacter pylori, commonly known as H. pylori, is a type of bacteria that can significantly impact stomach health. It is a major risk factor for developing stomach cancer. The journey begins when H. pylori colonizes the stomach lining, leading to a condition called chronic gastritis. This persistent inflammation can cause a series of changes in the stomach tissue, moving from non-atrophic gastritis to atrophic gastritis, then to intestinal metaplasia, dysplasia, and ultimately gastric cancer. One of the primary ways H. pylori contributes to stomach cancer is through chronic inflammation. When the bacteria infect the stomach, they trigger an ongoing inflammatory response. This inflammation damages the stomach lining, leading to cell death and tissue injury. Over time, this environment can promote genetic mutations and cellular changes that may result in cancer. H. pylori also has specific virulence factors that directly affect gastric cells. One of these is the cytotoxin-associated gene A, often referred to as CAG-SAE. This protein is injected into stomach cells, disrupting their normal functions. CAGA interferes with cell growth controls and enhances cell movement, which can lead to uncontrolled cell proliferation and cancer. Another important factor is the vacuolating cytotoxin A, known as VAC-A. This protein causes cell damage by creating vacuoles, inducing programmed cell death, and altering immune responses. These actions contribute to tissue injury and the potential for cancer development. The infection also affects the genetic stability of gastric cells. H. pylori can impair the cell's ability to repair DNA damage, leading to an accumulation of mutations. These mutations can activate oncogenes, which promote cancer, and deactivate tumor suppressor genes, further driving cancer development. Moreover, H. pylori infection induces various cellular stress responses. These include endoplasmic reticulum stress and oxidative stress, which can help some gastric cells survive and grow abnormally, acquiring malignant characteristics. Environmental and lifestyle factors also play a role. Smoking, high salt intake, and the consumption of processed meats can increase the risk of H. pylori-related stomach cancer. These factors may enhance bacterial colonization or amplify the effects of virulence factors like CAG-SAE. From the perspective of ulcers and peptic ulcer disease, H. pylori is well known as a primary cause of peptic ulcers. It damages the stomach lining and disrupts the protective mucosal barrier. The chronic inflammation and cellular damage caused by H. pylori that lead to ulcers also set the stage for the progression toward gastric cancer, especially if the infection remains untreated. In summary, H. pylori causes stomach cancer through a combination of chronic inflammation, direct bacterial effects on gastric cells, genetic damage, and altered cellular stress responses. This progression often follows a sequence from gastritis to precancerous lesions and finally to cancer. This highlights the importance of early detection and treatment of H. pylori infection to reduce the risk of both ulcers and gastric cancer. Always consult with healthcare professionals for personalized medical advice and treatment options.